Well, thank you for the introduction, and thank you to the organizers for giving me this opportunity to talk, and also to you to give me an opportunity to be heard as well. So my name is Antoine. I'm going to talk about the comparison of automatic and manual approaches for phage genome annotation. So as I'm sure you're very well aware of, phages are a very good opportunity to control multi-drug resistant bacteria. But they need to be very carefully selected if we don't want to spread unwanted genes in the environment. And for this, we have to study their genomes. The thing is, if you download a random phage from GenBank, you're going to have 70 to 80% of genes of unknown function in your genome. And so the poor quality of phage genomics and lack of tools has been noted by several authors. But the issue of quality in bioinformatics goes far beyond our field of phages, as demonstrated by this quote from Thank you. So let's say someone publishes a mistaken annotation in a phage in a gene database. So that annotation is susceptible to be used by another researcher who might then propagate that mistaken annotation in the database, and that's what we call database poisoning. Basically, a first mistake that is taken without peer reviewing by several papers. But then working well in bioinformatics isn't just important for the sake of science, because I think it's important for the field of phage therapy as well. Because if poor data is used in a phage therapy project, that project might be at risk. It will probably take slower to complete. It's going to be more expensive and have more chances to fail. That's going to be a very negative signal to funders and regulators in a field that is very fragile right now. And with less money, phage projects are more likely to fail. So bioinformatics practices impact science, industry, and in the end, patients. But everything is not so dull in the field of bioinformatics for phages. Anyone can quickly annotate a phage using Pharoka. If you don't like command line interfaces, you can use Galaxy. You can even submit a protein sequence to Collabful and get a 3D protein structure prediction. And you can also get excellent training in the forms of the CPhage program. So the CPhage program is led by Graham Hasfull Group at the University of Pittsburgh, and it aims at promoting student interest in science and in phage therapy by having them isolate and characterize their own phages. So at some point, they are offered with the opportunity to manually annotate phage genomes. And for this, there are obviously given protocols that you can consider like as the golden standard for phage genome manual annotation. And so they've been publishing more than 5,000 high-quality genomes, but the only downside of those protocols is that they take a very long time, and so they're not suitable when you are trying to build quickly a large collection of phages that you want to characterize as well. So Rime Bioinformatics is a project that aims at promoting a safe advent of phage therapy for patients and the environment. The project has 4 pillars. The first one is software development. We also have a combined expertise of phage therapy, genomics, and regulation. All of this is built on a solid ground of quality procedures in order to be compliant with the demands of pharmaceutical industries. And we have also a strong engagement towards our mission, so to help bring safe phage therapy. And we also help academic and private projects with their bioinformatics. So the tool we developed is called Rime Tools 2. And so to go back to the basics of gene annotation, it works on a very simple principle. You have a gene of a known function, then you're trying to find its function. You compare it to genes of known function that are in a database. And if you find a gene that is similar enough to the gene you're trying to annotate, you can consider that you have a likely function for that gene. As a researcher, when you're trying to choose the database that you're going to use, you have the choice between small curated databases that are going to give you high quality results, but since they are so small, the chance to have a match between the database and the gene you're trying to annotate are fairly slim. Or you can use large uncurated databases with much higher chances to have a match, but you don't really know the quality of that match. So our method relies on 2 technological bricks. One is a set of large curated database and a sensitive and stringent querying method, which allows us to have more results without compromising the quality of those results. And we also strongly believe in manual intervention, so our pipeline is made to allow expert manual curation at every step to make sure we don't let an automatic mistake propagate. So we claim that our tools 2 is able to annotate 60 to 80% of an average phages gene. And so I was presenting some of our results last year at the Evergreen conference when I met Kieran Furlong from the University of Ottawa, who happens to be a teacher in the CPhage program at the University of Ottawa. And he asked me that question: can automatic genome annotation compare with manual approaches? So we decided to find out, and Kieran gave us 27 phage genomes that were annotated by CPhage students, and we re-annotated them using the automatic version of our tool, and then we compared the results. And here are those results. So let's start with structural annotation, which is basically gene calling. So you can see that we missed on average 1.5 genes per genome that were found by students using the CPhage protocol. But for functional annotation, we were able to find 4.2 genes per phage genome on average that were not found initially. So to go back to Kieran's question, can automatic genome annotation compare with manual approaches? Yes, but manual curation is still necessary. The good news is we're already working on it, and we are planning on releasing the next version of our tool at the beginning of next year with improved annotation for phages and also for bacterial genomes, while staying regulation compliant, ISO 9001 and GXP compliant. So we still have a lot of work to do, but if you're interested in working with us to help promote a safe phage therapy, come and talk to us, and we'll be happy to see what you can do inside or outside bioinformatics. Thank you.